Thank you very much. So, thank you very much for coming this afternoon. One of the nice things about writing one of these books in the series, the very short introductions, is that it's a chance to have an overview of the subject, and you can delve everywhere. What you do find is suddenly, as you go outside your own area of expertise, you're actually learning in the way that the reader would be learning. And there is an enormous amount of interesting work that I've squeezed into the very small number of words in the book, bringing us right up to date. But my own work is on smell, and I wrote this book on animal behaviour and pheromones, which is all about communicating by smell. And these are the invisible signals between animals of the same species. One of the things that surprised me when I was writing that book is that as you go across the animal kingdom, everything is by the sense of smell. It's not by taste, it's by smell. And it's by those amazing noses that you see as you go across the animal kingdom. So it's detected by the sense of smell, and it really is the key to behaviour right the way across the animal kingdom. Now, I get to talk about pheromones just a few times in the book. I realised that I had to cover the whole of animal behaviour. But it's a special year this year because it's the 60th anniversary of the identification of the very first pheromone. And this was in the silk moth. Budendant was a chemist in Germany. He'd already won the Nobel Prize for identifying the structure of the human steroid hormones, the first of the reproductive hormones. And he then spent with a big team 20 years working out the first pheromone. And the problem with pheromones is that the animals concerned are incredibly sensitive. So only a few molecules are needed to excite the male moth. But that makes it very challenging for the chemist because the quantities are tiny. And he made a very wise choice. He chose the domesticated silk moth, and that allowed him to get 500,000 moths, because that was the number he needed to get 11 milligrams of material. The other thing that was great about it was he worked out how he could use the male moth's behaviour to tell him when the pheromone was present in his solutions, because it was completely invisible and beyond the chemistry of the time, but he could ask the moths. So what he devised was what we call a bioassay. So there's going to be a female moth coming in across the screen, and there's a male there. He's motionless until the female arrives, and he detects the molecules of pheromone that are coming from her pheromone gland, and they're pretty feeble, but eventually he will find her. But this allowed him to track the presence of the pheromone and ultimately to synthesize it and then go back to the male moths and say, have we got the female sex pheromone right? And it's the 60th anniversary this year. So there's extraordinary evolutionary pressure, selection pressure, on the male moths to be the first one, and they evolved these extraordinary antennae, almost like molecular sieves, to track the molecules as they fly upwind. Now, since 1959, pheromones have been identified right the way across the animal kingdom. If you're intrigued about where we are with humans, I did a TED talk, and basically the answer is disappointing. There could be a strong placebo effect of spending $50 and getting the pheromones in the post, but actually there really is no evidence at all. But they work underwater in goldfish and crabs, possibly even in birds, but you can probably spot your favourite animals there. Now, I said that I had to cover the whole of animal behaviour in the book, and I want to go right back to the beginning of human observation. So when did we start to watch animals? And I think the answer is probably as soon as we were becoming human. And there's a bit of evidence for that. If you go to the cave paintings in Lascaux in France, you find these beautiful paintings complete with the handprints of the artists. Now, not shown on this particular photograph, but on others, you can see a male cattle, a bull even, sniffing at a cow who's in estrus. And it's just the same kind of behaviour that you can watch on David Attenborough now or on a farm. So I think as soon as we were hunting animals, as soon as we were domesticating animals, we were interested in animal behaviour, because that would allow us to catch them, but also to look after them better. The first written records are with Aristotle, and in his voluminous account of zoology, he includes all sorts of behaviours. He talks about the ways that bees arrive if you give them sugar water, and bees appear from nowhere. Once you have one bee, lots of other bees will follow. He talks about parental care in catfish. He talks about the way the nightingale learns its song, and that's actually still a model system. So he was prescient. He got lots of things wrong, but he was the first person to record animal behaviour that we have records of. By the time you get to the 19th century, our observations are actually going more towards anthropomorphism. So people are writing about dog behaviour, and it's all about how the dog is feeling sorry for biting the furniture. And it's really not telling us very much about what animals are doing. An exception is Charles Darwin, who describes the behaviour of honeybees, not so much the dance, but the way that the hexagonal containers in the honeycomb must be designed by the bees, and how their behaviour must have evolved to produce those. So Charles Darwin takes characteristically a much more analytical approach. But where it starts to move into a generally scientific approach is at the end of the 19th century through into the early 20th century, and we have the development of what we would call now comparative psychology. So these were early observers of animal behaviour, but it was comparing the abilities of animals between species, trying to understand human behaviour and psychology in the context of what animals were doing. And one of the very first was Charles Turner. He was particularly interested in insect behaviour. He was the first person from an Afro-Caribbean background to get a PhD from the University of Chicago. And one of his early papers was investigating colour vision in honeybees, published in the Biological Bulletin. And basically he solves the argument, the debate. He says, yes, there is conclusive evidence that honeybees do see in colour, about 20 years before von Frisch showed the same in Germany. Another early pioneer was Margaret Washburn, and she wrote the first basic textbook on comparative psychology that went to 3 editions. But these pioneers have tended to be forgotten, and in their place we tend to think of Pavlov with his conditioned reflex in the dogs in response to the bell. They start salivating in response to that stimulus. And of course Skinner with the Skinner box. And it's quite typical that he is wearing a white lab coat, because by the time Skinner's approach is taking over, psychology, instead of being broadly comparative and looking at honeybees and ants and dogs in the wild, it really has become a lab study. So one of the early presidents of the American Psychological Association joked that the journal of the society was basically the journal of pigeons and rats, because basically those were the animals that were being studied at the time. So there was a movement in the 20s against that, to actually go back to watching animals in the wild. And this was the field, they coined the word ethology, and in 1973, 3 pioneers of ethology were awarded the Nobel Prize. Lorenz von Frisch with his honeybees, and Tinbergen. So the first of those was von Frisch, and here he's looking at bees that have arrived at a sugar water station, and they're going to be going back to the hive to tell their nestmates to come out to find the sugar source. And the thing that he noticed was something that had been noticed before, but he actually demonstrated that this really was a proper signal. And what the bees are able to tell their fellow nestmates back in the hive is the direction to fly in relation to the sun to find the sugar source, usually, of course, in nature, the nectar, a patch of flowers, but also by the duration of the waggle run, how often the bee waggles as she shows this in the hive, how far the bees should fly. And this is an arbitrary signal. The bees in Egypt waggle for a different period of time to indicate a certain distance. There was a huge controversy in the 1960s where some scientists said that the bees were not doing anything by way of signal with the dance. All the message was from the scent that the other bees were picking up. And finally, that was actually shown to be the case, that it really was a directional signal giving both the direction in relation to the sun and also the distance to fly. The second Nobel Prize, the person sharing it, was to Konrad Lorenz, who's famous for his work on imprinting. And one of the illustrations I've used in the book is this sketch by Nico Tinbergen, who visited from Holland, where he was based, to Konrad Lorenz's field station. And that's Lorenz in his +4s. And basically, he's attracting the goslings who've imprinted on him rather than their mother, who they ought to be following. So basically, the goslings imprint on the first moving object they see after they've hatched. So Tinbergen again took questions into the field, but what he was very good at was asking questions and devising simple experiments to try to tease out what might be going on. So the question here was, why did the gull parents remove the eggshell? And this is a very characteristic behaviour. Once the gull chick has hatched out of the egg, the parent picks up the eggshell and flies a short distance and drops the eggshell. So, why were they doing it? Well, what Tinbergen hypothesized was that the white inside of the broken shell would be conspicuous to visual predators. So, he wondered how you could test that idea, whether the crows and larger gulls, black-headed gulls, would actually find the white eggshell. So how could you test the idea? Well, he didn't have enough gulls' eggs to test it, so he painted chickens' eggs, hens' eggs, in the camouflage pattern, and then he would take an intact egg and then a broken egg with the white inside of the shell showing, and he placed the broken eggshell at different distances from the intact one, and then wait a few days to see how often the whole egg was taken. And the answer is, the closer the broken shell is to the whole egg, the quicker the predators find it. So he was able to show that it does seem indeed to be the function of the behaviour, that the parent picks up the broken eggshell after the gull chick has hatched and takes it away so that the visual predator doesn't find the rest of the eggs that have not yet hatched. Now, usually we can't see evolution happening, but sometimes in animal behaviour we can. And this is some work by Marlene Zook in Hawaii. And what she noticed over a few years is that the crickets on the island she was working on disappeared. She also noticed that she couldn't hear any crickets. And even as the crickets came back in numbers, there was no longer any cricket song. So Ed Yong, one of the science journalists on The Atlantic, called this the silence of the crickets. So what was going on? Well, the answer turned out to be this fly. This fly is a phonotactic fly. It basically will fly up to a loudspeaker that is playing the song of the male cricket. And what was going on was revealed when, if you look away now, it's a gruesome slide of the maggots inside the male cricket. So this poor cricket has been parasitized by the fly. She's laid her eggs on the cricket, and if you open up the cricket, it's basically full of the maggots. When those pupate, the females of that fly will then go off and find new male crickets. So what was the explanation for firstly the disappearance of the crickets, but then the return of silent males? So the answer, of course, was that there had always been rare mutants who never really found very many mates because they couldn't sing, because their wing was malformed. So they would only rarely find females, but there were still mutations every now and again. But in the presence of the fly, only those males survived. So the population rapidly became dominated by those males. And so that's why we have the silence of the crickets. Now, I mentioned that you can get parasite takeover, and we have zombie cockroaches and zombie ants. And this is now an enormous field within animal behaviour, because what you can use these natural experiments to do is to actually work out what's going on inside the brain of an animal. And what often happens is that there are precise injections of neurotoxins to different parts of the brain, just in the same way as your dentist would direct an injection if she's doing some root canal treatment. You can actually understand a bit more about what's going on. Now, in a longer talk, I discuss all sorts of examples of this, including Toxoplasma, which might affect humans and make us more reckless. But the example I want to talk about today is a wasp, and it's an example of where the wasp, rather than the postman, always stings twice. So, this is the jewel wasp, and if you Google or look on YouTube, you'll find lots of amazing video of this very fast-moving, beautiful wasp tracking down a cockroach. Now, these cockroaches move fast. This is the big American cockroach, and if you've ever tried to catch one, you'll know it's really hard. But these wasps manage it. They sort of creep towards it and then jump, and they bite, and whilst they're grabbing onto the side of the cockroach, the pronotum, she then brings her abdomen forward and stings, and she makes 2 stings. So the first sting is to the walking centres in the ganglion. So in insects, their nervous system, the central nerve cord runs down the front on the underside. So her first sting, which basically prevents the cockroach from moving, it basically momentarily paralyzes it, and that allows her to do a precision second sting. Now, the second sting, this is a painting. She basically is going in through the soft tissue of the neck, and then she's probing around inside the brain to find the right place to make her injection. So it's very much the same when, if you've got a good dentist, they don't have to do the injection very often. This wasp is amazing. So, what the scientists realized was that the sting can be coming in from all sorts of different angles. So it's actually really quite an interesting problem for the wasp, because she has to put the injection in the right place, but she won't always be coming in from the right place, because she'll have grabbed the cockroach, which she's having to do in haste, at various places along its pronotum. So the researchers first looked at where the venom was going. So they used a radioactive venom, a radioactive label in the venom, and showed that there were particular places that it was going. And they were able to look at the scanning electron microscope pictures of the tip of the sting, and they were able to show there were mechanoreceptors. So as she's feeling in, she can actually get feedback to her brain as to where she is, a bit like having doing keyhole surgery when you've got a video camera so she can see what she's doing, but she's doing it by touch. And she'll also have chemosensors, so she'll also be able to know, as it were, by the taste. And the researchers did something really fun, which is they started to look at what were the cues that the wasp was using to work out where to put the sting. So what they're timing here is the sting duration in minutes to the two stings. In red, the head sting; in black, the thoracic sting. So in the control, she's quick. So in less than 30 seconds, she's making the first sting, and it's taking her less than a minute to do the head sting. If you remove the brain altogether, that doesn't affect how quickly she gets the thoracic sting, the first one. But basically, she takes 10 minutes, and she knows the brain must be there somewhere, but she can't find it because the experimenters have cruelly taken it away. If they put soft gel, soft agar there, then basically she's pushing, but there's nothing to push against. So again, she takes about 10 minutes. But if they put a small pellet of hard agar, which is about the same consistency as the brain, basically she thinks that she's found the target, and so she gives her injection. So the nice thing about this is the way that you can dissect the behaviour and work out what the cues are that the female wasp is doing. Now, where does this relate to neural circuits in the cockroach? Well, one thing I didn't mention was the second injection does something really weird. The first thing is that it basically sets the cockroach off cleaning her limbs, and she's basically totally distracted. So it's a bit like putting butter on the paws of a kitten. Basically, they're distracted and don't run away. But what it also is telling us that the cockroach no longer moves, but can be walked to a burrow that the wasp has prepared, just like a self-propelled lawnmower. And the reason that works is the ability to start walking has been switched off by that second injection, but not the neural circuits for walking. So once the wasp tugs, the cockroach can walk, and those automatic circuits will start going. Now, those kinds of circuits that are automatic are highly conserved across the animal kingdom. It's the same kind of circuit that works in vertebrates, like us, to control breathing, and for that matter, walking. It means that we don't have to think with every step. And those circuits are really interesting. So one of the first things, though, is when a cockroach is still, it's not the same as when it's being stung by the wasp in the head. So the first cockroach was just resting. If you touch it with a paintbrush, it walks and it runs. But the second cockroach has had that sting, so that it actually will no longer properly walk until you actually tug it along. So it's actually quite different. It's not just being silent. But the reason it's interesting is one of the times that walking stops working is when you have Parkinson's disease. And there are problems with dopamine. And one of the things about this head sting venom is it's full of proteins that are part of the venom. We don't know which are the ones that actually are doing the work, but basically this is providing some drug leads. And whenever you see this in a press release, it actually means that in about 10 or 20 years, they'll have found a drug. So it was very exciting, but actually we're a long way off. So what about another topic? Collective behaviour is a big area in animal behaviour. It's turning out to be very exciting indeed, and it's all about many individuals working together. So if you have one common starling, it looks like this, and they are beautiful iridescent birds. If you have a murmuration of 10,000, it looks something like this. And you may have seen these, they're prominent over Brighton. This is a film from the Netherlands, and they are just truly extraordinary. So how do they do this magical moving in tandem, in fact in sequence, in synchrony? Well, some of the work has been done over Rome, where they have a really big problem. And there's some beautiful footage of this in the David Attenborough programme about animals living in cities. And some physicists took stereoscopic photos of the flocks of starlings, the murmurations over Rome. They digitized in 3D, and then they modeled the behavior. And it seems that what the starlings are doing is looking at the birds, about the 7 or 6 birds that are around them, and following them. So it's actually an immediate response to the closest birds. Now, why is that useful? Well, the first thing you have noticed about the film of the starlings is that the density changes. So sometimes they're very close together, sometimes they're far apart. Why does it matter then, the mechanism that they're using? Well, the answer is that one of the reasons they're doing this flocking may be to avoid predators. And if you are looking at many, but with very low cohesion, then there's a chance that when the hawk comes in, you'll be split off from your flock, from the murmuration. If you're tracking other animals, no matter how far away they are, then you'll always stay in a coherent group. And that might be the reason that they're doing it this way. But one of the nice things about this is that many of these examples can be modelled in very simple ways. So the final story is about one example, perhaps two, of applying animal behaviour. And the first is about elephants and bees. So the African wild bees are very vigorous defenders of their nests. And even elephants are scared of them, because elephants have sensitive spots around their eyes where, although the rest of their skin is thick, the honeybee sting can get in. So some researchers noticed that the elephants were avoiding the honeybee nests that were avoiding the acacia trees that had nests in them. And so they reasoned that it could be that the elephants were avoiding the bees. So the first thing they did was do playback experiments of angry bees, the buzzing of angry bees, and it turned out, yes, the elephants ran away. And moreover, they had an alarm signal so that if one elephant heard the bees and started running, they would give the rumble of bee alert, and all the other members of the family would run too. So the next idea was to see whether this could be useful in resolving some of the conflict between farmers and elephants. And the problem with that, with using playback of the songs of angry bees, is that the elephants would very quickly realise that there weren't any real bees, that actually it was a loudspeaker. So it turned out the solution, and this was done in some experiments collaboratively between the scientists and the farmers, was to set up beehives around the allotments, and the beehives are connected with a wire. And if the elephants creep in, and try to sneak between the beehives, it triggers the wire that shakes the beehives, and the bees come out and sting the elephants. So it actually works to keep them away from the crops, but it's also providing another source of income, because you've got honey and beeswax also coming from those beehives. So if you'd like to learn more, there's much more in the book, of course. On BBC Earth, you can find a fantastic collection of videos. If you Google TED pheromones, there's a very short talk about armpits and worse. And if you'd like to know more about the zombie ants and the fearless mice, there's a talk I gave at Gresham College earlier in the year. And if you want to follow me on Twitter, it's all there. So thank you very much indeed.
My question is back to the wasp and the cockroach. Why does the wasp do that to the cockroach?
So why is the wasp doing it to the cockroach? It's because it's a highly evolved relationship, all to the cockroach's disadvantage. So basically, in other species of related wasp, she simply lays her egg inside the cockroach. In this case, she lays her egg on the outside of the cockroach, but she has to keep it alive for the whole period that her wasp grub is feeding on the still living cockroach. So yes, it's much worse. It was before 9 o'clock, so I couldn't show you the rest. It's just really gruesome. And it's actually one of the things that Darwin wrote about in The Origin. And it's one of the things that almost causes him to question the hand of God. How is it that these really bizarre and unpleasant relationships have evolved between these parasitoid wasps, these ichneumoniid wasps, and their poor insect hosts? And there are in fact, there are more parasites than there are animals hosting parasites. And one of the reasons for that is the wasp is a parasite on the cockroach. There is almost certainly at least another wasp that's smaller that parasitizes the wasp. And there may even be a hyper parasite that parasitizes that wasp. So basically, whenever there's an unused food resource, a parasite evolves to parasitize it. And it's just a very good way of living. The cockroach has to spend its days searching for food and running the risk of being eaten and all those other things. By taking a fully formed food object, that cockroach, putting it in a secure burrow with her egg that will hatch into a larva, she actually provides for her offspring in a beautiful way. So there's all the benefit to her. So that's why parasitism is such a profitable way of living.
How long is the cockroach the host?
For about a month.
Okay. And then it just dies?
It's basically a husk by the end, yeah. And often the videos show the beautiful jewel wasp emerging from the pupa next to the husk of the cockroach.
Thanks. Sorry, what was the comment you were going to make about Toxoplasma in humans?
So Toxoplasma, this is a brain parasite, and people studying rat behaviour realised that the rats that were infected by this brain parasite, it's related to malaria, but in this case it's actually lodged inside the brain, inside neurons and glia inside the brain. It changed their behaviour profoundly, and instead of being scared of cats, they now approached cat urine and cats. And so there's a beautiful set of papers which are all about fatal attraction. So it looks as though in rodents, the infection by the parasite changes their behaviour. And the story goes along the lines of the parasite needs to get into the cat for the next stage in the life cycle. So what the parasite is doing is changing the behaviour of the host, the rat or mouse, so that it is more likely to go to the next stage in the cat. So people then discovered that humans often show traces of infection. So basically it leaves an immune trace, antibodies, in your blood if you've been infected. And there are some beautiful studies showing that if you look at army recruits in the US with a very big sample and look at the exposure to the parasite, it does look as though those recruits are more reckless. They get involved in more vehicle crashes. And there was some Czech work that suggested something similar about they were more likely to be run over, the people were more likely to be run over and appear in A&E if they had an infection. There was even a suggestion that it might be involved in schizophrenia, that it might be some kind of relationship between infection with the parasite in the brain and changes in human behaviour. The problem is that the story is one of those that I wish was true, but may not be quite as neat as it sounds. So as you go around the world, the infection rate varies enormously between different countries. So in some countries it's at about 2%, in others at about 30%. But for example, schizophrenia rates don't follow that at all. And of course, as you'll know, schizophrenia is incredibly difficult to identify, to diagnose, and they may have multiple causes in any event. And when people have gone back to the rodent work, even that work turns out perhaps it doesn't always give the simple story. So it's one of those intriguing things that it may well be true. From my own point of view, I think it's one of those that's sort of unproven. In Scotland, they have the verdict, is it not proven? So you have guilty, not guilty, and not proven. And I think in this case, it is one of those. It's one of those stories that you just wish was true. And it really is fascinating. But there's a big literature on it, and it's worth searching out. But it's just one of thousands of examples of parasites apparently manipulating the behaviour of their hosts. So thank you.
I was wondering if pheromones were detectable between species and the sort of the scope for confusion that creates.
So are pheromones detectable between species? And the answer is yes. And it's in part because any broadcast signal can be sniffed, can be detected by something else. The same as a Wi-Fi signal can be sniffed and picked up somewhere else. So there are lots of examples where predators have receptors that are just as sensitive as their prey to the prey's own pheromone. So one of the things in California was in the 1960s when people were working on bark beetle pheromones. So in California, the bark beetles attract other members of the same species to overwhelm the defences of a tree. And basically, if all of them land on the same tree, they overwhelm the tree. So the researchers at UC Davis and UC Berkeley were trying to work out what the molecules were. So they put them out in the field, and they were expecting to catch bark beetles. What they found was, again, about 600,000 predators who were homing in on the bark beetle pheromone. And when later Hanne Mustapata in Norway looked at the receptors, those receptors on the predators were just as sensitive as the receptors on the prey. So you can eavesdrop. It actually gets even more twisted. There are spiders that illegally, as it were, synthesize the pheromones of their prey. So they pretend to be female moths, and they release the pheromone at night, and they, instead of having a web, the web is contracted into a sticky ball, and they swing it a bit like a gaucho, and they're called bolas spiders, and they detect the males flying upwind, and when the male gets close, they flick the sticky bolus, catch the male wasp and draw it in. So you can have deceit in both directions. You can have eavesdropping by illegal receivers, but you can also have illegal production of pheromone, and you can have a very good living that way. So very good question, thank you.
Thank you for this, by the way, really interesting. I'm always intrigued by the influence that animals and humans have on each other. So looking at like dogs or cattle that essentially we have created, I guess, which animals have had the biggest influence on human evolution, be it unwitting or wittingly?
So which animals have had the biggest effect on human evolution? Yes, so...
Predators, either way.
Yeah. And that's a really good question, because usually we tend to think about domestication in the other direction, but they are domesticating us. I think the biggest change is lactose tolerance. So basically, in Northern Europe, there was selection for the ability to continue digesting lactose from milk, so that's the milk sugar, after weaning. And so we're very good at it. My husband, whose parents came from the Caribbean and ultimately from West Africa, he's lactose intolerant because in his ancestry there wasn't selection for lactose tolerance. And so he's on soya milk, whereas I can drink regular. So there is an effect, but that's not so much on our behaviour, apart from choosing soya rather than regular. I'm not sure. There are some nice suggestions that there's a big group in Hungary who's looking at the interaction between dogs and humans. And the suggestion is that compared with wolves, dogs really do look us in the eye. So there is a real communication. So wolves are not bothered if we're pointing, but puppies and adult domesticated dogs will follow the pointing and take that as a signal. Now, whether they've had an influence on us to point, they certainly have had an effect on telling us when it's time to feed them. So when I go and stay with friends, the dogs and cats all get very active at about 9 o'clock, and they suddenly become very friendly because it's 9 o'clock is the time that they get fed every evening. So in that sense, in the short term, there is definitely an effect on our individual behaviour. Longer term, I don't know, but your question is a good one, because every evolutionary interaction between species goes both ways, so there must be. So thank you. Yeah.
Hi, thanks so much for this talk. Going back to the lactose, why do humans wean in the first place?
Why do humans wean? Well, that's something actually that you see right the way across mammals. The thing that defines mammals, of course, is producing milk. And it's a very energy-intense food. It's a way of getting your young through a vulnerable period until they can really handle solid food. In some animals, how long that takes varies from species to species. But at a certain point, because producing milk is also very energy intensive, if your young are actually able to do the digestion themselves or whatever food your young feed on, then it's time for them to stand on their own 4 legs and start feeding. So in every mammal species, there is a moment when the mother, in effect, tells her offspring, time to do it on your own. And she starts to dry up, her hormones change, she stops producing milk. And this has been investigated in some detail in terms of the interaction between the mother and the offspring. So Patrick Bateson, over many years, studied the weaning behaviour in domestic cats and kittens. And basically, there is a moving away and eventually a pushing away by the mother, because the kitten would actually still like the milk. But it's time for the kitten to start eating solid food. And it's the same in sheep. So the mother willingly feeds the lamb, and then at a certain point, it's actually time for the lamb to start feeding on grass. There is also a link to a much more theoretical question, which is all about the question of what's to the benefit of the offspring and the parent. So it's a big area in animal behaviour called parent-offspring conflict. So from the point of view of the kitten or the lamb, or for that matter, a human baby, it's all about them. So for them, what they want is milk for the longest possible time, but for the mother, it's at the cost of future offspring. So at a certain point, she needs to stop feeding this generation, or rather this litter, and start to keep her resources for the next time she's going to produce a litter or a baby. And so that's another reason for the conflict. So it's not just that the lamb or baby or kitten should be on their own 2 feet, it's also that there is a conflict between the interests of the reproductive success of the mother longer term and this particular kitten. So it's actually a very interesting area which has been studied in some detail now. Thank you.
My question is, do you have a favourite animal and why?
So do I have a favourite animal? No, and that's not just like being a parent. Do you have a favourite child? No, I'm actually fascinated by animals of every kind, and I still find myself amazed. It was actually Tim Bergen who I never met, but it was his books that got me into animal behaviour. He had a book for young adults called Curious Naturalists, and it was all about his work in the sand dunes on wasps and then later on the gulls. And I was just basically hooked. And over my career, I've worked on fish in the Red Sea, on octopus, on insects, on moths, beetles. I've never worked with every kind of animal. I've never worked with elephants. So there are lots of animals that I would love to work on. But one of the things you also realise is it's actually sometimes much better watching them on film. Because one of the things about animal behaviour that they don't tell you is it's really boring. Because what you see on David Attenborough are the edited highlights that they've been waiting 3 years for the belly flopping blue whale or whatever. And all those times they've been bitten by mosquitoes in the jungle if they're looking at some other behaviour. And what you see is the best bits. So I don't have a favourite animal, but I actually just love watching them. So no, thank you. Do you have a favourite one?
Dogs. Probably dogs. That's a
Oh, well,
good
dogs.
one.
Yeah.
All those funny little wasps now.
Yeah.
They're cool.
Yeah.
So I wanted to ask, why would what kinds of communication or messages are pheromones good for, as against just sound or visual communication?
So what kind of messages are pheromones good for as opposed to sound and other ways of signalling? The answer is especially at night. So one of the things that pheromones can be used for is particularly nocturnal animals. So that's one reason that moths perhaps use them, because they're flying at night and finding the female. It's very good for long-distance communication because so long as the molecules can get carried by the wind or the water downstream, there's almost no limit. And one of the things about visual signals is that you need to be in line of sight. And for sound, depending on the habitat, that attenuates very quickly. One of the problems with pheromones in the wind or water is that it's very hard to locate the source. And so animals have evolved all sorts of neat ways of navigating, orientating upwind, and that's a whole area of study in itself. One of the interesting things is that you have multimodal signals. So the humble fruit fly uses both sound and pheromones and touch and taste for its signals. So sometimes it's a pheromone, sometimes it's sound, sometimes it's all of them. And one of the fascinating things is to relate the use of pheromones to particular habitats to particular species. And sometimes it's almost traditional in an animal group. So if you're a moth, female, you tend to use a pheromone. And that's really reflecting the phylogeny, it's reflecting the evolutionary history. But that whole question about when do animals use pheromones, when do they use other modalities, is a fascinating one. So thank you. Yes.
Thanks for the talk. It was really, really interesting. So talking a little bit about human behavior, in your talk you've discussed setting up controls and lab experiments and so on. And given how that's not possible for humans and also the longevity of human, I guess, lifespans, what are, I don't know, some of the ways we can study human behavior by analogy to animal behavior, or from, you know, sort of the challenges in that area?
So learning about human behavior from animal behavior. I think in a way that basic question underlies so much of the interest in animal behavior. Whether that's model systems for Alzheimer's in mice, where you're looking to see whether or not those tau proteins are doing something in the brains of mice. One of the problems, though, is that even going between mice and rats, it turns out they're very different. And what doing experiments, doing observations on animal behavior, what those experiments and observations can tell us is always with the big caveat, with the big question, how does it apply? But it is the kind of thing, so long as you are very cautious, and good scientists are, it's always of interest. So you can, particularly in higher primates, see things like affiliative behavior, the friendships between different animals in a group, the way that conflicts are resolved, all sorts of different behaviors can, I think, tell us things. The problem is the wild speculation. And the book I didn't write is one of those many books that has a little bit of animal behavior and then suddenly 20 pages of speculation that this means, that this means. But it is the ever interesting topic because we've always watched animals, and whilst we're watching animals, we're trying to see if that gives us an understanding of how we behave. And I think in the way that Darwin always recognised, we are simply another animal. And I think we've got all sorts of things that other animals don't have. The list of things that are super special to humans keeps on diminishing, whether that's different kinds of language, tool use, almost anything you can name turns up, well, there's an animal that does something similar. So all the behaviours that animals do probably do have some clue to what we do as humans. So yes, thank you very much.
Brilliant. I think we're at time now, but thank you very much, Tristram Wyatt.
Thank you very much. Thank you.